basically wait downstairs near the building. And the second is, you have noticed that, okay, there are two museum visits. One is today and one is tomorrow. So museum is just here, basically. So this is the some collection of C.V. Raman. So both the days the visit will be there, and I think he can accommodate about 25 to 30 people at a time in the museum. So just choose one of the day, and you basically have to just come out there and just go to the end of the corridor and then open the door, and that's the museum basically. Yeah. So first is a group photo for, okay, whoever has come now. So we'll just have a group photo immediately after this session. So you just wait downstairs. Yeah.
Good, thank you. I think we're going to start. The first speaker of the session is Joachim Krug from the University of Cologne.
Okay, yeah, thank you. And I'd also like to start by thanking Sanjib for and his team for putting together this very nice meeting. I'm very happy to be here after some years of absence from India and other places, and to see so many familiar faces and friendly faces. So I want to tell you a little bit about work that we have been doing over the last years on modeling biological fitness landscapes, particularly with the emphasis on evolutionary accessibility and the effect of structure and fitness landscapes on accessibility. And I'll sort of explain what I mean by these terms as I go along. And this talk is based on a kind of review-style paper that I posted on arXiv last week. So, you know, this if something that I cannot cover, you can sort of look it up here. So I'd like to start actually with a schematic, a kind of bird's eye view on evolution, which goes back to a talk by Amitabh Joshi, a colleague of Kavita's at JNC, that I attended when I was visiting Bangalore for the first time in 2015. And so Amitabh told us that, you know, we shouldn't be afraid of biology. Biology is basically very simple because there are only three phenomena. And these phenomena are development. Development is the process by which an organism is sort of created or appears out of its genetic information. So it takes on its physical appearance, which we call the phenotype. Then there is a phenomenon of ecology by which these organisms interact with each other and with their environment. And this then determines their reproductive success, which is sort of what we quantify in terms of the fitness. So think about fitness as the number of offspring of an organism in a certain environment. And then, through the phenomenon of heredity, this fitness sort of couples back to the genotype, because the reproductive ability determines to what extent the properties of the organism, the genetics of the organism, will be represented in the population in the next generation. And as you can see, this forms a kind of feedback cycle, and this is really the engine behind Darwinian evolution, right? So this is sort of what gives rise to the entire complexity of the biosphere. Now, the concept of fitness landscapes that I want to discuss today is a kind of conceptual shortcut in this diagram, where we say, okay, since in the end what really matters is how the genotype determines fitness, let's suppose we can sort of model that directly, right? So and of course you can see that this is an extremely bold simplification, because you're basically throwing away almost all of biology, right? Nevertheless, it turns out that this concept, it's almost 100 years old now, and it has sort of stood the test of time, and it's becoming sort of more of more current interest again with recent high throughput methods. I'll show you some examples in a moment. Yes?
Is fitness landscape strictly in terms of fitness landscape?
No, you can do a mapping here as well, of course. Yeah. In principle, you could also map the phenotype. I mean, actually, at the end of the talk, I'll show you, you know, a model where we sort of represent this two-step process approximately. But so here, I'm always going to think about mapping genotype to fitness, right? And this is what Wright was doing. So these are two images from his 1932 paper, and they already contain the sort of the most important elements of the theory. On the one hand, you have the sequence space. So this is the organization of the genetic material. In modern terminology, we would think about the space of all DNA sequences, of all proteins. Wright at that time was thinking about the sequences of genes with their different variants, which are called alleles, but it's still the same structure, and I'll talk a lot about the structure of the space as I go along. And so Wright's idea now was that each node of this graph is a genotype, so a particular sequence, and you assign a fitness value to each of these nodes, right? And Wright's intuition was that these landscapes would be rather complex. There would be many peaks. So the idea now is that evolution drives you to a peak of this fitness landscape. But of course, if there are many peaks, then this is a complicated process, just like we know from physics, from systems with complex energy landscapes. And so this is sort of the second image from Wright's paper, which is a kind of topographic map showing peaks and valleys. And Wright thought that an essential problem that theory would have to face would be how evolution navigates this landscape, right? So the navigability of these landscapes since then is sort of on the agenda. Now to make this precise, let me describe the mathematical setting that I will be using in this talk. So genotypes are sequences. They have a certain length L. And the letters in the sequences come from an alphabet of size A, which can be 2 or larger. And these different symbols here are generally called alleles, right? So these are just the choices of a symbol that you can have at one site. There's a natural distance measure on these spaces, which is the Hamming distance. So you just measure the number of sites at which the two sequences differ. And then a fitness landscape is simply a real valued function of the space, right? So we have a map that maps each sequence to the real numbers. Now, generally one assumes that if each of these symbols can mutate to any other symbol. This doesn't have to be the case. I'll get back to that later. But if that is the case, then essentially all sequences that are at distance 1 from each other are neighbors on the graph. And then we can think about path on this landscape in terms of path of genotypic sequences where the distance between two consecutive elements is 1. And then the notion of evolutionary accessibility that I will be using here is simply that fitness increases along the path, right? So I'm just going to ask, I'm going to say a certain genotype is accessible from another one if there is a path of increasing fitness going from one to the other. So again, in the physical analogy, think about an energy landscape and ask whether a zero temperature dynamics would be able to take you from one place to the other, right? So most of the time I will be using the binary alphabet, not always. And then for the binary case, we can think of the 1 as being the presence and 0 being the absence of a certain mutation. So we just consider at each site, we consider one letter and one other letter, which is then the mutant. Okay, so then let's sort of illustrate this. So this is a small L equal to 3 cube of sequences, and so here I've sort of imagine having fitness values on these nodes. So now I've sort of illustrated them by the sizes of these circles, and in order to study this accessible path, it's useful to sort of orient the arrows in the direction of increasing fitness, and this then makes this into an oriented graph, which we call the fitness graph. Now in this particular situation, so suppose I want to go from the all 0 string to the all 1 string, so I have to add these 3 mutations, and I can do that in 6 different ways, right? There are 3 factorial different orders in which I can add the mutation. And here you can see that along all these paths, all these 6 paths, fitness increases. So here, all these paths are accessible. And such path that takes sort of the minimal distance between 2 points, I will call direct path, right? So here, all the direct path from the 0 string to the 1 string to the peak are accessible. Now we can sort of modify this fitness landscape a little bit. So now I've put a larger fitness here. So now you see we have a kind of global fitness maximum here, and we have a local fitness maximum here. As a consequence, out of the 6 direct paths that we had before, 2 now become inaccessible, because if you want to go here, you get stuck at this maximum and you cannot go any further. But now there are in addition additional paths which are indirect. So for example, if you want to go to this local peak, then you can use an indirect path that goes here, or you can use an indirect path that goes here, right? And there's one direct path as well. So you see that as you sort of make the landscape more complex, you have, you might get more indirect path that can get you where you want to go. So the questions that I want to explore for this talk is, so first of all, and this is sort of the, when you see a picture like this, or in particular when you see the experimental pictures on which this is based that I will show you next, the natural question for a statistical physicist is, how many paths should you expect if the fitness values were completely random, right? So that seems like a useful null model, and this is a question that we formulated more than 10 years ago, so that's sort of what I want to cover in the first part of the talk. On the other hand, of course, it's clear that real fitness landscapes are not completely random, so now suppose that the fitness landscapes have some structure, ideally biologically motivated structure, how will that change their accessibility, right? So these are the sort of two leading questions that I want to discuss here. But let me first then show you a couple of empirical examples. And so the paper that got a lot of this recent fitness landscape work started is this one. So this was published by Dan Weinreich in 2006, and it has this very nice title: Darwinian evolution can follow only very few mutational paths to fitter proteins. So this paper is about a particular enzyme, an antibiotic resistance enzyme that hydrolyzes a certain class of antibiotics and thereby confers resistance to bacteria. It's a very ancient and very important, medically also very important enzyme. And so Weinreich was studying 5 particular mutations in this enzyme that were known to increase the resistance against a certain antibiotic by a big factor. And so this is what you see here. So this is a part of the sequence space. Here the notation is, so the minuses are what I call zeros and the pluses are what I call ones. So this is sort of the original enzyme, the so-called wild type, and these numbers here are measures of resistance. So essentially you measure how large the concentration of the antibiotic can be so that the bacteria can still grow. And you see that if you go from this point to here, you increase resistance by a factor of about 45,000. And so what Weinreich did was to sort of map out all the intermediates, right? You have 5 mutations, so you have 2 to the 5 different combinations, so you can sort of generate them and you measure the resistance for all of them, and then you can sort of identify the pathways along which resistance is increasing, and this picture is showing the most important ones. And there's some notation here, so these are sort of typical notations for protein mutations where you, at position 42, you place an A by an G and so on. And then they evaluated the mutational pathways. So since this is a system of size 5, there are 120 different direct mutational pathways, but in fact only 18 out of those were found to be accessible, right? So this is sort of what gave rise, despite the fact that this landscape is single peaked, right? So there's only one peak, but nevertheless a lot of these pathways are not accessible. So that was sort of the message of this paper. In a somewhat later publications, they also looked at indirect pathways, and then, so this is more or less the same picture. And here you now see a couple of pathways that are indirect. That is, you first add mutations, and then you go back. So in this case, these indirect mutations include so-called back steps, where you decrease the number of mutations. And that increases the number of pathways a little bit from 18 to 27. But at the same time, of course, the total number of pathways is now enormously much larger, because this is just the total number of self-avoiding path on the 5 cube, right, which is this big number. So you have an enormous increase in possible path, but in fact only a small number of paths that actually become relevant. So this is now by now quite sort of classic work. And of course, since then, the experimental methods have evolved enormously. So now people are studying fitness landscapes on completely different scales. And this is an example from last year, which covers a problem that is very close to all of our hearts, the SARS-CoV-2 protein. And you might remember, it seems like a long time ago, at some point there was this Omicron variant that appeared, right? And so what this group did, this is a group of Michael Desai, is that they studied, so it turns out there are 15 mutations that separate the Omicron variant from the ancestral Wuhan strain. And this group, what they did was to create all the combinations, right? So in this case, there are 32,000 combinations. And then they measured the affinity of these intermediates to a receptor in the human cell that is necessary for virus entry. So this affinity is a kind of, if you will, a kind of proxy for the ability of the virus to enter the cell. What you can see is that on average, actually, the affinity goes down. So this is a kind of broad fitness valley that the virus had to overcome. And in fact, one believes that it did overcome it because it was better at evading the immunity of the immune system of the human. So it didn't spread because it had higher affinity, but because it had better ability to evade the immune system. But for the present context, what I just want to emphasize, so these authors also checked for accessible path, and they found that there's not a single accessible path in this landscape.
Just to make sure, accessible means where every step is fitness increasing?
Yes, yes.
So it strongly depends on what we mean by fitness.
Well, whatever you measure, affinity. I mean, we have some number, right? So I have some number, and I'm asking, does this number increase? I mean, in practice, of course, people always measure proxies of fitness, right? So here the fitness proxy is this resistance measure, just a concentration, and here it's an affinity, right?
And that comes back to also the question of Martin. It seems to me intuitively for the little biology I know that fitness is a very complicated
Of course,
question,
yeah.
also by ecology and phenotype.
Sure, yeah.
So if you just by just knowing the genotype, it seems you have very little to speak about fitness, but it's just because I don't know that probably.
No, but in principle, of course, I mean, there are many caveats to this, right? But in principle, if the environment is fixed, then if you could hold all the conditions fixed, there would be a genotype fitness map, right?
If everything would fix.
And of course, in reality, the systems that people look at are, very restricted. So this is clearly, I mean, that that the affinity of a protein is determined by its sequence, I think, is not so difficult, right? I mean, I think that one can believe, right? All right, okay, so, and I'll actually show you another recent study later where actually fitness is measured in the sense of growth rate of a certain variant in a population, right? So in principle, you can also measure these fitnesses. Okay. So now let's start with the question of random fitness landscapes, right? So let me take this space, right, and put random numbers on it. And so this leads to a problem that we have called accessibility percolation. So there's another review article here that appeared in a book that you can look at, which has more details. And so the first important thing to realize, and maybe I go back to the schematic here, is that accessibility is determined only by the ordering of the fitness values, right? So this fitness graph is determined by which fitness is larger than which other. So it's an order statistics property. So in that sense, the distribution from which I draw my fitness values actually doesn't matter, right? So if I just keep the same order, this will give the same result. So the distribution isn't important, but for convenience, for the following discussion, I'm just taking these fitness values to be uniformly distributed random variables. So now we can ask, what is the probability to find a fitness increasing path? So let's look at two genotypes, sigma and tau, and let's suppose their fitness difference is some. So the fitness difference here will always be denoted by beta. This is some number in between 0 and 1. Obviously, the fitness difference has to be positive because otherwise there cannot be a path, right? Now, the condition for a path of length L to be accessible is that all the intermediate fitness values have to be in the interval between the end and the beginning, and they have to be increasingly ordered, right? And so putting this together, we see that the probability for a path to be accessible is this. And now we want to, so the problem that the quantity that we want to study is the number of accessible paths between two particular genotypes, right? So this is a non-negative integer valued random variable, and we're particularly interested in the probability that this random variable is greater or equal to 1, right? So that there's at least one path. So this is what I define as accessibility. And the question that I want to focus on, and this is sort of where this name accessibility percolation comes from, is the existence of a threshold so basically it's clear that if the fitness difference is large, it will be more likely to have an accessible path than if the fitness difference is small. And so I'm particularly interested in paths that are sort of macroscopic length, if you will. So the distance between the two genotypes should be of order L, and I can scale it. So then I have this delta, which is just the relative distance. And I want to know, as a function of beta, is this probability non-zero or not, and does it change at some threshold beta c? And that's then what we call the accessibility percolation threshold. So you have an onset of accessibility at a certain fitness difference. Capital L is just the size of the sequence length. So the sequence length is like the maximal distance, right? So it's the diameter of the space, if you will, right? Okay, so the first rigorous study of this was done by Hegarty and Martinson, and they considered direct path on the binary hypercube. Now, if you have, in the direct case, as I discussed before, the number of paths is of length L is L factorial, because it's just the number, the order in which you do the different mutations. So the expected number of paths is just L factorial times the probability that it computed before, which is L times beta to the L minus 1. And so you see that as long as beta is less than 1, this will vanish asymptotically for large L, right? Beta is the fitness difference between the 2 points, okay, within this uniform distribution. Okay, so the expectation value goes to 0, and then, of course, by Markov's inequality, you can sort of bound this probability by the expectation, and so this probability also goes to 0. So you see that if beta is less than 1, then you will not have any direct path on this hypercube. Now, to show, so you might expect if you put beta equal to 1 that you would have such a path. So for beta equal to 1, you see that the expected number of paths grows linearly in L. But of course, you cannot really, you cannot just conclude that, because you have to, the Markov's inequality only works in one direction. So what you have to do is to analyze the fluctuations. So you have to analyze the second moment. And so what Hegarty and Martinson showed that indeed, if beta is equal to 1, the probability of having an accessible path is equal to 1. And in fact, more precisely, if you allow if you look at a sequence of beta values that are approaching 1, if this approach is faster than log L over L, then you will have accessibility with probability 1. And this scaling you just get from demanding that this expectation value should be equal to 1. Okay, so in the directed case, we can say that this hypercube is sort of marginally accessible. That is, you will have a chance of finding such a path only if the fitness difference between the beginning and the endpoint is maximal. Okay. Now let's look at indirect path. And this was first studied by Berestitsky, Brunet, and Xi, and later rigorously proved by Martinson and Lee. So indirect path means that you now have paths that are longer than the direct path. So let's again look at the 3 cube. So this would be a direct path from the all 0 to the all 1 string. This would be an example of an indirect path, and this is another one. So you see that the length of an indirect path always has to be, there has to be pairs of backsteps, because each backstep you have to sort of revert, right? So if you have p backsteps, then the length of the path is 3 plus 2p. And so this is like the longest path that you can have. So in principle, in order to, in principle, the approaches could be, you could use the same approach as here. That is, you just take this probability and you multiply it by the number of paths. The problem is that the number of paths is very difficult to evaluate, and in fact, you don't know it for general L. But Berestitsky et al. Were sort of creative, so they were nevertheless able to compute the asymptotics of the expected number of paths. So this is again what we want to focus on here. So this is again the expected number of paths, and I'm taking L to infinity. I'm taking this to the 1 over Lth power, and then Berestitsky and collaborators showed that this limit is equal to this expression here. So this delta is the distance, right? So beta is the fitness difference, delta is the scale distance. And so, again, you have so this can be less than 1. If it's less than 1, then you know that there are no paths because of Markov's inequality. If it's greater than 1, there could be a path, right? And so when this is equal to 1, this is sort of your candidate for a threshold function, right? So beta c of delta would be the solution of this equation. So Beresitsky et al., they sort of conjectured that this would actually give the true threshold. So they say what their terminology here is that they say the expectation tells the truth. And this was actually subsequently proved by Martinson and Lee. So by just looking at the expectation, you can actually predict where this transition occurs, right? Yeah. No, a backstep just means that you decrease the number of ones, right? So here if you have, okay, let me show this. It's an indirect path, right? So let me show this one again, right? So here, so you add one, you add another one, you remove one, right? So this is sort of what a backstep means. It just means that you're not following the orientation of the cube, right? Fitness always increases. I mean, this is sort of what our condition is, right?
Fitness doesn't necessarily depend on the number of ones.
Sorry?
Fitness doesn't depend on the number of ones.
No. Right. So here only the length of the path matters, right? Okay. So this is and so this gives you this gives you a solution, and so in the sort of worst case where the distance delta is equal to 1, this gives you a beta c of about 0.88, right? So now you have a kind of true transition. So for fitness value, fitness difference is smaller than 0.88, you don't have a path, and when it's greater than 0.88, you have a path. Now, in recent work with my student Benjamin Schmiegelt, we were able to generalize this, these considerations to multi-allelic fitness landscapes. So now you don't have the binary hypercube anymore, but you have A different alleles on each side. Biologically relevant cases, of course, are the DNA and RNA, where you have 4 letters, or A equal to 20 would correspond to proteins. Now here it becomes important, especially when you look at the example of proteins, it becomes important that in general not all alleles can be turned into all other alleles, right? So not all transitions between different symbols here are actually possible in one mutational step. So therefore we have to introduce another kind of structural element into the model, which is a so-called mutation graph. So you can basically define this graph by an adjacency matrix. And so that just tells you whether a transition between a pair of alleles is allowed or not. And I'll show you some examples in a minute. Also, the specification of the initial and endpoints of the path becomes a little bit less trivial. I mean, in the binary case, all that you have to know is how far these 2 points are away from each other. But here you also have to specify, what kind of symbols there are in the sequences, in the endpoint and initial sequences. So this we can formalize by another matrix, which is again, so these are both sort of A times A matrices. So this is another matrix which tells you the fraction of sites at which the initial point has a K and the final point has an L. Okay? So that's sort of just a way of specifying, if you will, the direction in sequence space. Yes?
In this mutation graph, only one thing can be changed at a time, or you can even have 2.
No, only one at a time. Yeah, you can only do one mutation, but some mutations will not be allowed. So I'll show you examples in a minute.
E going to infinity, will that be the NK model?
We'll get to the NK model. You'll see. All right. Okay. So for this, Benjamin is an extremely good mathematician. So he was actually pushing, and after significant efforts, was able to formulate the result rigorously. So this is actually a theorem. And so basically, the theorem is that, similarly to here, that in most cases the expectation gives you the right threshold. So just putting the expectation equal to 1. Now the expectation in this case, and this is not too hard to understand, can be obtained by taking this exponential of the adjacency matrix and here taking it to the pKl power. So this is just a function of this matrix A and the matrix pKl. So, and this you can in principle compute for any mutation graph that you like. And so when this is equal to 1, so this will have a solution, or will have a solution beta star. You can show that this function typically has a single zero, a single, or the log of it has a single zero. So this function has a single solution, which is 1. And so here I wrote a bit sloppily that this is true for most but not all mutation graphs. So it turns out there is a condition on the mutation graph that is sort of difficult to explain, but I'll show you an example of a graph that doesn't satisfy this. So in general, this beta star will be a lower bound on the threshold because of Markov's inequality again. But in most cases, it will actually be the exact threshold. Of course, if this beta star turns out to be greater than 1, then there will be no accessible path, right? Because you cannot have a fitness difference in these units that is greater than 1. So these are a couple of examples of mutation graphs. So this is sort of the standard nucleotide mutation graph, right? With 4 symbols, the 4 letters in the alphabet. So for example, for this one, we have an explicit formula for the beta C, which is given by this nice equation. So it's about 0.5. So if you remember in the binary case, the threshold was 0.88. So the threshold goes down. So that tells you as you increase more, as you increase the alphabet size, the landscape becomes more accessible. This funny looking graph of size 5 is the smallest graph for which this condition that I haven't explained to you is actually violated. And the last example is a so-called path graph. So this is a case where you basically, you can only mutate these symbols in a particular direction, and then in a particular order. And then it turns out that the beta star is greater than 1. So in this case, again, there wouldn't be any accessible path, right? And so this is another sort of biologically important example. This is the amino acid mutation graph. So because of the structure of the genetic code, you cannot mutate every amino acid into every other one by one nucleotide mutation, right? So this is sort of showing which mutations are possible there. So in principle, you can also evaluate this expression for the amino acid mutation graph. And this is just a summary of results for the complete graph. So if I'm assuming that the mutation graph is complete, that is all alleles can mutate to all other ones, then this threshold at maximal distance delta equal to 1 can be expanded for large A, and the leading order term is log A over A. So that tells you that as you increase the number of alleles, the landscape becomes more and more accessible. And this is sort of showing so this is delta and this is beta. So this is showing the behavior for different A's. This is a binary case. And so you see that this sort of goes down. So basically what I'm saying is that 2 genotypes at this distance and with this fitness difference would be accessible from each other if they are above the curve and not accessible if they are below the curve. One can also actually compute the path length. So it turns out, so you could also ask, these indirect paths, how much longer are they than the direct path? And this is given by a factor of about log A. So it's a kind of moderate increase in the length. Okay, how much time do I have? Okay, so this was the random situation, right? Now, of course, as I said, yeah.
So if you go to the Weinreich's example, and if they had actually kept track of all the nucleotides, you expect more paths? Maybe they have that data?
If they had tracked, that's a good question. I guess so, yeah. I guess. Possibly, yes. I mean, there have been experimental studies, that have shown that these indirect paths actually are important, right? Well, I'm looking at, one needs to do more, right? So the expected number always gives you a lower bound on the accessibility, right? But to actually prove that this is also an upper bound, you need to work, you really need to look at more higher moments and so on. So there's a whole story here that. Not well. You know, you can produce estimates that are good enough to prove this, but it's not. Yeah, you know, you can look at this paper. Right. Okay. So real fitness landscapes, of course, are not random. They are structured. Right. So this is sort of the extreme case of a structured fitness landscape. You know, a landscape which just has one peak, and there, of course, you know, basically the peak is accessible from anywhere, right? So wherever you start, you can always find a path to the peak. So the question is, if you now introduce this kind of structure to a landscape, to a fitness landscape model, what does that do to the accessibility? And so a classical way of doing this is Kaufman's NK model that was already mentioned here. So very briefly, what you do in Kaufman's NK model is that you write the fitness as a sum of contributions, and each of these contributions is just a random function of a subset of the sites, right? So for example, if these subsets are of size 3, this would be an example, or of size 2, and then you have to you have to sort of specify how you choose the subsets. You can organize these. You can organize the sites on a ring, or you can you know, subdivide them into disjoint sets or arrange them randomly. So this is a way of interpolating between the sort of perfectly accessible, namely single peak landscape, and the random landscape, right? Because if your subset is of size L, then you're back to the random model. Now it turns out that although these, you know, with smaller k, these landscapes have less peaks and are sort of less rugged and everything, they're actually not more accessible. So on the contrary, there are basically no accessible path for large L for any k that you keep fixed. And this is, again, work by Benjamin Schmiegel that is sort of reviewed in this paper. On the other hand, so this is not, you know, in some sense, not good news. On the other hand, experimentally, there are now examples of landscapes that are rugged, that have many peaks, but are nevertheless easily navigable. So this is an experimental paper from the group of Andreas Wagner that appeared in Science a couple of weeks ago. So they look at a large data set of about 260,000 combinations. So they look at all nucleotides at 9 positions in a certain gene in bacteria that has a certain role, again, in antibiotic resistance. And then they actually do fitness measurements. So they actually grow the bacteria, all these different variants, in the presence of an antibiotic. And so then they get a landscape. So of course, a lot of these combinations don't work, right? So the bacteria don't grow. But there are at least 18,000 sequences that are functional. So this is a big landscape. And for this landscape, they looked at fitness peaks. So they found about 500 peaks, and 73 of them have high fitness. So this is shown here. So the high fitness peaks have a certain amino acid at one of these positions, either a glue or an asp. So this is shown here. And now the remarkable feature of these peaks is that they all have very large basins of attraction, right? So you have these many peaks, and the basin of attraction here simply means that there is a path from from a given genotype to the peak, right? Directed or undirected, we don't care about that at the moment. So you see that these high peaks have, you know, a significant fraction of the whole space is sort of is able to access them. And so most of the path, at least for up to a certain length, most of the path are actually accessible. So this is, and so in this paper, the authors make the point that this is sort of contradicts most of the models, and this is not, you know, it shows that we don't really understand the structure of fitness landscapes. But now it turns out that we have actually, so in work with, this is sort of work spearheaded by Suman Das, who until recently was a postdoc in my group, and, you know, I want to mention him here particularly because he's a graduate of RRI, so he graduated here in 2015. He's now in Bern in the group of Claudia Bank. And so so here we came up with we sort of stumbled upon a model that actually does combine high ruggedness and high accessibility, right? So this is sort of schematically illustrated here. So this is sort of what you would expect. You have a landscape with one peak, and it's accessible from everywhere. Now you have 3 peaks, and these are only accessible from part of the landscape. But what we really need is a model that has combines these two things. And these models are characterized by a particular property that was discovered by Suman. which is called the accessibility property. Now, to explain that, it's useful to introduce a different notation. So we can identify a binary genotype with a subset at which the symbol is 1, right? So in this way, the all 0 string would be the empty set, the all 1 string would be the whole set, and so on, right? So this means that you can have subset superset relations between the genotypes, and so then the accessibility property is the following. So we say that a fitness landscape has a subset superset accessibility property if any peak is accessible from all its sub and supersets along all direct paths. So this means that each peak sort of spans a kind of a basin in which there's essentially all the paths are accessible, right? This implies in particular that there's a lower bound on the basin of attraction because if you have a peak that has n mutations, that is, the set is of size n, then this set will have 2 to the n subsets and 2 to the L minus n supersets. And if you then subtract the set itself, you get this a basin of attraction that is of this size. So these basin sizes, grow exponentially with a number of letters, right? Now you might worry that these landscapes are not very rugged, but that's actually not the case. They are typically also, as rugged as let's say the NK model. So they have exponentially a number of peaks that grows exponentially. And in fact, you can using Sperner's theorem, you can prove that there's an upper bound on the number of peaks, but this upper bound is very large. So this is of order 2 to the L over square root of L. So there isn't really any limitation on that. So how can you generate these landscapes? Okay, so I think I'll skip over this one. Okay, I think I don't have time to explain that. So maybe I should show this picture instead. So this is sort of an illustration of the accessibility property for a landscape with 2 peaks, right? So here, this is one peak and this is another one. And so all the red ones have to belong to the basin of this peak, and all the blue ones have to belong to the basin of this peak. There is a simple way of generating such landscapes that I maybe show here. So this is the way in which you can easily generate these landscapes. And this sort of comes back to the beginning of my talk. So here we're sort of putting in a kind of phenotype into the picture. So we first map the genotype into a phenotype, which is just the real number here, which is just a linear combination of the sigmas. And then we map this phenotype onto fitness through a function phi, okay? And then we can prove, it's actually a simple proof, but I don't have time to explain it, I think, that this landscape will satisfy the accessibility property if this function phi is concave. I mean, the point is that the accessibility property, again, can be proved from a certain inequality, which we call universal negative epistasis, which is a relation between the fitness effects of genotypes that are subsets or supersets of each other. Okay, that we sort of came up with in this paper. So in principle, there's an easy way of generating these landscapes, and then you can study things like, for example, the distributions of the sizes of basins of attraction. So this is an example. This is a model, a landscape of size 8, where these coefficients here, so we choose these coefficients here at random, and we choose them from an exponential distribution with mean 1. And the fitness function is a simple negative parabola, a nice concave function. And this shows, and so now the bound that I showed you on the basins of attraction sizes depends on the number of mutations. So this is shown here as a red bar. So these are the peaks with 1 mutation, 2 mutations, and so on. And these are the distributions of the basin sizes where you either only count directed path or you count all path. And you see that there's a lot of basins that are quite a bit larger. Now, for the model for which this property was originally discovered, this is a model that requires a bit more explanation. This was a model that was designed to describe the evolution of antibiotic resistance at various drug concentrations. And this is the distribution of the basins of attraction sizes. And here it's sort of conspicuous that you actually have a lot of basins that are of maximal size, right? So we think that in this model, there could actually be, a significant number of peaks that are essentially accessible from the entire space. Okay, so I cannot explain how this comes about in more detail, but you can read it in the paper that I mentioned in the beginning. So let me summarize. So there have been spectacular advances on the experimental side, and that has sort of rekindled the interest in the structure of these landscapes. Somewhat surprisingly, if you introduce structure and in the landscape, that doesn't necessarily make it more accessible. But I think in particular, sort of the organization of the basins of attraction of these fitness peaks is an interesting question that we want to study in more detail in the future. And so let me just thank a couple of people. So Christina Krona, we collaborated with on this negative epistasis business. Suman, I mentioned. Daniel, Jasper, Stefan, and Benjamin have been master students, and Muhittin Mungan is a senior researcher in my group. And I should also acknowledge the interaction with Arjan de Visser and his lab, who has been sort of taught me everything I know about population genetics and experimental evolution. Okay, thank you very much.
So why do you insist on every step should increase the fitness? One can have bad mutations also, right? In general.
Yeah. I mean, this is a question I always get. I mean, it's it's a very restricted definition of the model. The advantage of this definition is I don't have to speak about the evolutionary process at all, right? So of course, I mean, and maybe Kavita will do some, explain more about the actual dynamics of evolution. But of course, so if you have an evolving population, of course, deleterious mutations can fix or they can play a role, so fitness doesn't always go up. But all of that will depend very much on, the population size, the regime that you're in, and so on. So you would need to specify much more, right? But this is a property that only depends on the measured landscape, right? So you don't have to, you get a picture of the navigability that is independent of everything else. Of course, to what extent an actual evolving population would go along these paths, that's like a separate problem. And in fact, even if there is a path and most of the paths are not accessible, of course, the landscape, the population doesn't know where the path is, right? So it might not find it. So, but that's like a separate class of questions, right?
How do the experimentalists access such a large number of combinations? You showed some 250,000 or something?
Yeah. Right. So basically, one approach is that, and I think that was what was done for the SARS-CoV-2 study, is that, well, first of all, you have to sort of generate all combinations, right? But that is, with sort of the mutational tools, I think that's not that difficult. So you have a pool where then of course, you have to somehow. Yeah, so let's, suppose you have generated a library that contains all the variants that you want, right? Then you can measure the phenotype of interest, in this case, the affinity to the receptor, right? For example, with a fluorescence assay, so they just measure some fluorescence intensity. And so then you basically bin your variants according to this phenotype, and then you sequence them, right? Then you look which variants are in which bin, right? And that way you can sort of get a map of this.
Some sort of automated process, or I mean.
Of course, yeah. I mean, it's large. I mean, yeah, of course. I mean, no, you will not get a student to pipette 262,000. No. All of this is highly automated, and of course, it's these are very complex procedures, and they probably have a lot of sources of error and so on. But as usual, you do replicate measurements, and if the replicates correlate well, you can be pretty sure that what you're seeing is real.
Okay, one more question here.
Does the accessibility depend on the mutation graph in any way, or?
Sorry?
The mutation graph. I
Yes.
mean, so does that affect the accessibility?
Yes.
So does it have to have a particular, form or something? No, what I mean is this this factor, this accessibility landscape or this accessibility effect that you saw? That
I'm not sure what.
No, I
part
mean the
you're
mutation
conferring.
graph was telling. It was
I mean
something
here,
that you
so
put
in the last part, the mutation graph doesn't play a role because everything is again a binary landscape,
Oh,
right?
I
So
see,
you
okay.
just go
Okay,
from one to the
I
other.
see,
So
okay.
so the mutation graph plays a role when you have more than 2 alleles, right?
Yeah, yeah.
And then it basically, you know, of course, the best you can have is like a complete mutation graph where every allele can mutate into every other. If you don't have that, then obviously that will restrict the accessibility, right? So so obviously you would expect that the complete mutation graph would generate the most accessible landscape, right?
Right.
And if there are some restrictions, then the accessibility goes down.
Yeah.
Good. Yes.
Is the interest in the number of accessible paths related to probability of acquiring the final mutated states or something?
So say it again?
So you have been looking at the number of accessible paths for a final mutation, right? Is it related to the probability of actually having the final mutation somehow? So?
Well, that would again depend on what kind of search dynamics you consider, right? You know, the probability of seeing a particular genotype in a certain population dynamics. Of course, I mean, intuitively, you would think that if you have, a peak with a large basin of attraction would be more likely to be seen. And in fact, I mean, so in this paper, they actually also, no, not this one, the experimental paper, they actually also run, so of course, what you can also do is to run like adaptive walk simulations on these landscapes, right? So you can sort of run search processes, and then you can ask how often you end up at a certain peak, and that would be, and of course, that should correlate with the size of the basin of attraction.
Okay, good. I think we should go to the next speaker. Thank you again. Okay, so our next speaker is Kavita Jain from the Nehru Center in Bangalore.
Where's the pointer? Okay. Thank you. Okay, so good afternoon. So yes, so first of all, I would like to thank Sanjeev and the team for organizing this meeting, and of course, many congratulations to RI on the 75th. So I'll speak about genetic diversity in changing environments, just continuing the theme that Joachim has already started. And I will talk about some work that we started with Sachin Kaushik when he was a PhD student with me. And we are continuing some questions with his postdoc mentor, Parul Johri. Both Sachin and Parul are at University of North Carolina. Okay, so instead of starting with the motivation, I will start with a model, simply because all of you are physicists. So I'll define this model for you. It's called a Moran process. The model is very simple. We are thinking of a population of n individuals. Out of those n individuals, there's one which is a mutant, which I'll denote by 1, and n minus 1 are the wild type, which I'll denote by 0. So I have just startingly, to begin with, I have this population. And once I have put in 1, 1, no more mutations are allowed for various reasons. Okay? And the fitness of the mutant is 1 plus S. So now, by now, you know what fitness is, thanks to Joachim. And S could be positive, negative, or 0. The fitness of the wild type, that is of the zeros, is 1. Okay, so that's the setup. And now I'll define the dynamics. It's a simple birth-death process. That is, a birth can happen here with a probability which is proportional to the fitness of the type. And the death is, however, equally likely. So, for example, at time t, if I happen to have these zeros, 4 zeros, and 2 ones, ones have a fitness 1 plus S. So this could give birth. It's chosen with a probability proportional to 1 plus S. And there are I of such mutants. Therefore, the birth, the probability that the number of mutants, so let me say it again. So if there are I mutants at some instant, then the number of mutants increases by 1, which is the probability proportional to how many mutants I have, okay, in the previous time step. And then because I've increased the number of population by giving a birth, one of them must die. And to make this difference, one of the zeros has to die. And since that's equally likely, therefore there is a factor of n minus I. Similarly, the number of mutants decreases by 1 if one of the zeros gives birth, because they have fitness 1, so that's proportional to n minus I, and one of the ones should die, and that's multiplied with I. Okay, so this is just this simple process. So probability with which I can become I plus 1, I can become I minus 1, and of course, there could be that 1 gives birth and 1 dies, or 0 gives birth and 0 dies. In that case, I remains I, and therefore sum add up to 1. So that's a simple model, which I will refer to again and again. Okay, so that's a simple model to keep in mind. Okay, so as you can see, and if you run this in your head, you can see that this model has 2 absorbing states, because I'm not allowing any mutations. Once all the individuals become 0, because 0 cannot become 1, or all the individuals become 1s or 0s, because we're not allowing any further mutations, system just stays in that state. Okay, so these are the 2 absorbing states in this model. Okay, so therefore one of the simplest questions that we can ask is, what is the chance that if I start with a single mutant, eventually all of them become mutants? So this is called, in this literature, it's called fixation probability. And this is something which physicists are very used to calculating. So what we can do is to simply write down a backward Fokker-Planck equation for the model that I described to you. But let me describe the terms in this Fokker-Planck equation. So if the population size is infinitely large, you have a deterministically evolving population, and the term that we in physics, which is coming with the, call it drift term, it is of the form S times P into 1 minus P. So what is it saying? It's saying that if S is positive, that means the mutant population is growing exponentially fast, but there are only a fixed number of them. There's a finite carrying capacity. As a result of that, I have factor 1 minus P. That's the meaning of this term. On the other hand, we also have this so-called diffusion term, and this is, of course, coming when you calculate the variance of the change in the transition probability, the change in the variance, so it's in the variance of transition probability, which says it's simply the binomial sampling. That's the meaning of this P into 1 minus P. The typical problems that we do in, of diffusion constants is a constant. That means when you toss a coin, number of heads, it does not depend, the number of heads in today's experiment, it does not depend upon how many heads I got yesterday, right? So that number over there is a constant, it's P and Q here. It depends upon these variables as well, because the number of offspring you produce depends upon how many were there in the previous generation. So these models are different from the usual simple models that we consider, in which these numbers are, these factors are constant. Okay, so this is the most basic model, and one can add other things to it, but this is the one that I'll think about. Okay, so now this is the backward Fokker-Planck equation, because we want to know what's the probability that eventually all become mutants. So that means if I start with the P frequency at time t, I want to know that the final frequency becomes x equal to 1 at a very large time. So therefore, dp by dt is equal to 0. You can work it out. And with these boundary conditions, if you impose, then one can find out this fixation probability. And it's given by the simple expression, which is known for a very long time. So let me explain this expression to you. It's saying that if the mutant is a good mutant, that is S is positive and is very positive, that is S is much, much larger than 1 by n, then the chance that all become mutant is simply S. On the other hand, as you intuitively expect, if S is negative, that means it's a worse mutant, then the chance that all become mutant is exponentially suppressed. In between, you have 1 by n, which you expect, again, from simply symmetry. Okay? So therefore, the chance of a good mutant to spread is substantial, whereas a deleterious mutation is simply very, very less, it's exponentially suppressed. Okay, so this is a simple fixation probability. And the question that I'm interested in today is called mean sojourn time. So what is that? Let me explain that to you. So I find it easier to think about it, first of all, in discrete space and time. So suppose I start with a single mutant. So this is the number of mutants in the y-axis, and x-axis has time. So suppose I start with a single mutant, and then it does this Moran process, and then maybe I want to understand how many visits it makes to 3. That means how many times the number of mutants become 3. In the red trajectory, they become 1, and then again 2, and then perhaps this trajectory is lost, and that's the absorption that happens. In the blue trajectory, 3 is hit twice again. Okay? So the sojourn time is simply in mean number of visits to a fixed site, starting from some initial value, which is also fixed, before eventual absorption occurs. It's easier to work with continuous space and time, and therefore, it's simply the mean time spent between frequency x and x plus dx, starting from a value p before eventual absorption occurs. And that one can write down easily. So if I write down again the backward operator, backward Fokker-Planck equation probability, so if I want to know that at some time 0, the frequency is x, and it's a non-absorbing frequency, it's strictly between 0 and 1, then it should get contributions from all the allele frequencies at all the times that came before 0. So this mean sojourn time simply is this integral. Okay. Okay. So in general, we have this backward Fokker-Planck equation. You would integrate it, and then you can see that the mean sojourn time is simply the Green's function, okay, which can be evaluated very easily and subject to these boundary conditions. The typical thing, I mean, many things, questions that you heard in this meeting are about absorption times or first passage times, right? So that mean first passage time to either boundary is simply the integral over the final value of this mean sojourn time. Okay? So I hope the definitions are clear. Okay. Okay. Fine. So the Moran process that I started in the beginning, so for that, we can easily calculate, I mean, this is known for a very long time. This Green's function, I mean, we can calculate, and typically we are interested in initially small frequencies. So therefore, the final frequency, which is larger than p, where p goes to 0, so that expression only I'm showing here. So it's simply given by this expression, but let me look at it again in some detail. So again, if S is positive, that means a good mutant, then this mean sojourn time is a U-shaped function, okay? And it's a decreasing function when S is 0 or negative. So this makes physical sense because you see, if we are starting with very low frequencies, so initially, most likely the mutant will get lost. So therefore, the mean sojourn time is high near low frequencies. On the other hand, if mutant is a positively selected mutant, then it can reach, I told you in the beginning, it can reach the other boundary, the upper boundary, and therefore the mean sojourn time will be high there. So that's why it's a U-shaped function for S positive, and otherwise it's a decreasing function. It decreases very fast for the negatively selected mutation. It's a very fast decaying function. Otherwise, it's a classical answer called 1 by x. Okay, so these are the answers for the mean sojourn time for the Moran process. Okay, so what has all this to do with the genetic diversity? So a simple model that I described to you. So now let me get to some little bit of biology. So suppose I give you this sample. So suppose I take 6 of you, and we do the genomic sequencing. And for humans, this is something called reference sequence, which is very well known. So as you know, in 2003, the human genome genome was sequenced, and after it has been improved a lot. So there's a so-called reference sequence, which says that at every base pair, there is, you look at 1,000 individuals, and which is a consensus sequence, which has the most likelihood, what is the most likely nucleotide there. So there's a certain reference sequence, and I take 6 of you, and take your genome is sequenced, and I compare it with the reference sequence. If, let's say sample number 1, if it matches at the first locus, if it matches the reference sequence, I put a 0, otherwise I put a 1, and similarly all of the rest. Okay? So I take the 6 sequences and compare them with the reference sequence, and I get this kind of data. So that's the kind of data one is thinking about. And the question that biologists are interested in asking is how many diverse loci there are. Diversity is incredibly important for evolution. If we are all identical, there's nothing going to happen, right? So we have to be different. So diversity is important here. So for example, the 7th locus, it may be different from the reference sequence. In this population, it's identical. So it's uninteresting. Okay, so this is the sort of question that people are interested in. And more fundamental question is, what kind of evolutionary forces shape this genetic diversity? Is it mutation, selection, stochasticity, or some other evolutionary forces? Okay. So the quantity, yeah?
This diversity is only at the
Yes. Today, I will do only genotypes. Okay. Okay, so one quantity which is a more detailed statistics that people calculate from this kind of data is called a site frequency spectrum, because it's maybe unfamiliar to most of you, so I'll write the definition right here. So I'll refer to this quantity a lot. SFS, site frequency spectrum. I'll do it in the continuous space and time. So therefore, this is the frequency, is the number, sorry. So it's the mean number of loci with frequency x at time t. Okay? So this is the quantity that people look at, and it's in more detailed statistics, and simply because it's of course calculable from the data. So for example, I can look at this first locus, see how many mutants you have, 1, 2, 3, and so on, and then make a histogram. So of course this is measurable from data, and it could be used to answer the questions about diversity. So if I don't care about how many number of mutants are there, I just find out, just sum over all this FJT, it will give me number of diverse loci, and similarly other statistics that people measure. Okay, so that's fine. We have all this data and so on, and we want to know what, diversity, how do we explain that? So therefore, one would like to model this. So there is this model which is used a lot in the subject, which says the following. So suppose let's look at this diagram. So what I'm plotting here is the number of mutants as a function of time. And what happened is, suppose a mutant came at this time 0 in this green trajectory, and it, say, followed Moran process or whatever, and then maybe it did increase, and then it got lost because I'm not allowing any more mutations, right? Then after some time, some stochastically, stochastic time, then another mutant arose, but this mutant arose at a second locus, at a different locus, okay? And then this is the red trajectory, which came again, and so on, it got lost, and so on, right? Okay? So therefore, we have, the assumption here is, in the model, that we are running many Moran processes, and all of them are independent. And each of the Moran process is starting at a different instant, which is stochastic. So at every instant, you figure out whether mutation would occur or not, and that rate is 2n mu. So at different instants, we are starting this Moran process, and then we are looking at this object SFS. Yeah? The model assumes that they're independent, simply because we cannot do anything more than simulations otherwise. Right, okay? Okay, so that's the assumption, and that's the model that I will look at. Okay. Okay, so what's happening in this model? So we have these trajectories which are getting lost because of absorption at either of the boundary, and the new mutations are coming in. So you can expect that perhaps some kind of stationary state is reached. So indeed, you can show, and I will see that equation later on. So there are trajectories which are going to be lost due to absorption, and there are new trajectories created due to new mutations, right? Okay, and therefore, when you ask in the stationary state at large times, how many number of loci are there with non-absorbing frequency at large times, it's this object, which is the mutation that created at one site, and then again, our Fokker-Planck probability. That is, at some time, it became X, and it will get contribution from all the trajectories that came before. So this is the time instant that I'm looking at. Then at this instant, the number of loci that have a frequency X, they will get contribution from all the trajectories that came before, right? Okay, so that's the quantity that one could write down. And this is the quantity we just saw. Because of the fact that these are all independent Moran processes, this object is simply proportional to the mean sojourn time. Okay, so this object of interest in the stationary state is simply proportional to the mean sojourn time. Okay, so but that's very useful in the sense that biologists use this to infer things about the population. So for instance, if they look at this data that I showed you, they will measure this SFS, and then if they see something like a U-shaped SFS, they infer that perhaps there's a positive selection in this population. If they see something decreasing, then perhaps it's 0 or negative, and then there are methods to infer what the value of S might be. just like we said that if I integrate over the mean sojourn time, I get mean absorption time. Here, if I integrate over this final frequency for the SFS, it gives me the mean number of diverse loci. And this is the simple integration you can do for this Moran process. And so there is a big factor of 2 and mu, which says the number of mutations produced. And then there is other factors of log, which is sort of the weakish factors. But the key point that is what this answer is saying is that larger the population, more is the diversity, which makes sense because, the largest population, more mutations can enter the population. Okay, and do keep in mind because I'll come back to this point. Okay, so that's all fine. What I just spoke to you is about equilibrium populations or the stationary state. But there's an interest in understanding this f of xt when, what is time dependence like? How does the diversity change with time? There's only very few, some work, but not very much. In some simple situations to slightly more complicated ones, there's some bunch of work. Surprisingly, perhaps, people are more interested, and in biologists are more interested in the cases when environment is changing with time. That is, the coefficients of our Fokker-Planck equations are time dependent, and especially if there's a demography. What it means is the size of the population is not fixed. It's changing with time, simply because it's a biological realistic assumption, and there's a lot of work done. So I'm showing these references since 2000 simply because of genetic sequencing. There's a boom in that in the early 2000s. So there's a lot of work when the population size is changing with time, relatively less when there's a changing selection, and with both factors changing, there's hardly any. Okay? Okay, so what we have is we want to think about a Fokker-Planck equation where these coefficients, that is of the first derivative and the second derivative term, they are time dependent. So I'm looking at that Fokker-Planck equation. And I'm again interested in this SFS. Okay? So it's useful for technical purposes. It's useful to look at now the forward Fokker-Planck equation. So now, if in general I have the population size, which is time dependent, so the mutation happened, and then this is the forward one. So therefore, again, we start with a low frequency P, and we want to know how many loci have frequency X at time T. So therefore, they get contribution from all the ones which came before, right? So it's just the same kind of definition, except for the forward Fokker-Planck equation. And now, the forward Fokker-Planck equation for this Moran process looks like this. And now you will notice that if I take the derivative with respect to time, I should get a delta function, right? Just like we did for previous case, because of the integration limit. Okay? So you can do that. You can put a delta function and you can work it out that way. But we prefer a model to work with in which the mutational input is modeled by a boundary condition. That means we want to solve this Fokker-Planck equation in which the boundary condition is such that at small frequencies, it has the mutation term, mutations came, and divided by x. This divided by x is simply because I gave you this answer of mean sojourn time for neutral case when their s was 0. That's simply because when we're at very low frequencies, what's important is only stochasticity. Selection and all, they're not important because the numbers are so low. So that's what the important thing, and therefore the boundary condition is that is used for this model. Yeah, so for this model is, the mutation input is therefore, mutational input is through this boundary condition, and then there's the other boundary condition. Okay, so how do we, generally, if I want to solve such problems, how would I do it? First of all, I have an inhomogeneous boundary condition, so first thing one should do is simply to shift things such that you have a homogeneous boundary condition, which one can write down. And then one would like to use the usual variation, the separation of variables method, and therefore I would write something like this. Selection is very important, but the problem with solving these equations is that when we write down these equations for selection, we don't get closed form eigenfunctions, for example. So for example, if I had this selection and mutation, etc., they were time independent. Even then, we don't know the solutions of, we don't know closed form solutions. So therefore, the eigenfunction problem, it turns out to be something called oblate spheroidal equation, which you can write down only as a linear combination of certain orthogonal polynomials. You can do it, but it's not very helpful. It's not very practical to work with. Okay? So that's a sort of problem in working when you have selection in the model. So when we have time dependence also, again, we get this 3-term recursion, which is pretty bad, like in the sense that we cannot really do much beyond numerics. Okay? The only case that which we know is solvable exactly is when there is no selection. And that's simply because it turns out the eigenfunctions we know exactly here for this operator, and it simply obeys the Gauss hypergeometric equation, for which we know connection formulas and everything. We can find an eigenvalues, so a whole lot is known, and therefore that's the only case which is exactly solvable. Okay, so I'm interested in these two sort of simple examples I wanted to give. One case that I want to consider is when the environment is changing in a periodic fashion in time. And the motivation is simply because there are seasonal variations which can affect the fitness. And similarly, demography can change in a periodic fashion, for example, as seen in predator dynamics. So, yeah. So we can, again, this is our Fock-Planck equation, and I'm assuming S and N are changing in a periodic fashion. In general, they can have different amplitudes, different frequencies, phase, and so on. The time scale that we have here, dimensionally you can see, are the size of the population, because that's where the stochasticity enters, the growth rate, and of course the environmental frequency, which I said for today equal to each other. Because, as I told you already, with selection it's not possible to do things exactly. You have to do something else, which I will not discuss. So therefore, I will just discuss a case. I will just discuss these approximations where the environment is changing slowly. By that I mean the environmental frequency inverse is much larger than the other two scales. And similarly, the opposite limit in which the environment is changing rapidly. Okay? And we'll be interested in SFS average over period. At large times, the Floquet eigenvalue will become 0, and f of xt is periodically changing, and therefore we can sort of do this average. And that's the quantity which we could look at. Okay, so slowly changing environment, let me just look at that. The simplest thing that you can do is to think of adiabatic approximation. That is, at each instant, the environment is changing so slowly that basically your population is in equilibrium with the instantaneous S and N. Okay, so you can do that, and if there's no selection, this is the answer that I showed you earlier. F of xt was just 2n mu divided by x. If you average, nothing happens because linear. Okay, but in this selection, then we have a nonlinear dependence on these parameters. So if S is positive and large, then we had this answer that I gave you earlier. And for the positive mutations, for the negative mutations, we had this exponential suppression. So when I average it, a trivial calculation shows that only the positive part of the cycle will contribute. Okay, so this is trivial calculation, but it has some meaning. So I want to describe that meaning to you. So what does it say? So it's saying that the answer for the time average SFS is 1 by x into 1 minus x. It's this non-monotonic function times this factor which says that if your population size is changing with time, then you only average when the S is positive, right? That's all it's saying. So suppose, imagine that this biologist has collected this data over a season, or over a year, and then they have averaged it over this full year. And they find that the SFS is looking like a U-shape, okay? And they might infer there's a positive selection, okay? However, you will produce this pattern of positive selection of a U-shaped pattern, even if half of the time cycle was positive and half of the time it was negative. That means on an average, even if selection was 0, still you will get this U-shaped pattern, and therefore you can misinfer the parameters. So people look for this kind of patterns, but if you assume constant selection, which is what is usually used, you may just misinfer the parameters. So that's why, that's a simple message from this little expression. There is this other brown part. This also has a meaning. Let me describe. So there's a major big problem in the subject called the Lewontin's paradox. So this was introduced almost 50 years ago. And the question is this. Suppose you have a simple theory which says no selection and so on, and then you calculate a diversity, and in this little model, you plug in the census population size. So for example, for humans, you put in a few billions, and you look at what diversity is. But actual diversity, which is measured, is much lower than what you expect from the simple theory. So for example, for humans, if you want to you measure the diversity and you want to check with the formula, you should put in a number like 20,000. So it's not 7 billion of us which are contributing diversity, just 20,000 of us. Okay? So therefore, what it says is, we do not understand why the diversity is so much lower than what we expect from some simple theories. And what I said to you earlier was, larger the population, larger the diversity. So if I'm seeing a smaller diversity, somehow the population size is smaller. And there's this concept of, therefore, effective population size, so which has all the other stuff might be going on, and that you introduce its effects within this effective population size. So here, I have introduced this as the effective population size, which includes the effect of changing selection and joint action of changing selection and changing population size, okay? And this is smaller than n bar, the average population size. Okay, so therefore it's sort of going in the right direction in the sense that the effective population size is smaller, and therefore diversity is indeed smaller than what you expect from naive expectation. I mean, you expect naively. Okay, one can also look at the rapidly varying environment, which is the opposite limit. In this case, we know the exact solution, and therefore we can calculate again f by x. Here, again, 1 by x, which is the canonical answer. And then again, you have an effective population size, which turns out to be harmonic mean of the changing population size, which is again smaller than n bar. Okay, and this is not nearly a new result, it's sort of expected, but as far as I know, this has not been shown before. When we introduce selection, we expect that when the things are changing very rapidly, the population will be sensitive only to averages, which seems to be true, but except not always. So, but I'm not going into detail of that. Okay, so to summarize, if we look at the diversity in the changing environment, so this is some other measure, as a function of the frequency of the environment, it seems to change non-monotonically. Whether it's a maximum or minimum, it depends on some other biologically relevant parameter which I have not described to you. So in any way, it sort of increases perhaps, or decreases then, or decreases first and then increases. It depends on some of the biological details. Okay. Yeah. Yes. Yeah. 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 It depends upon the frequency. The definitions work out to be different.
No, I mean, when you say qualitatively?
I don't know right now. I don't know. I have to think about it. Which ones? No. This is the averaged over a period. This is average over period. This is not periodic. It's just non-monotonic. Okay. Okay. So I want to describe one more model to you, and again, this is motivated by the Levante's paradox of low diversity, and I want to give you a sort of extreme case of this. So again, Levante's paradox is that diversity is lower than what we expect naively. So here is a mechanism which was introduced by Maynard Smith and Haig in the same time when the Levante's paradox came along, when Levante came up with his data. So again, let's think again as a physicist. Now again, I'm going to the Moran process, but this time instead of a single site, I have two loci. And these two loci are special in the sense they are physically linked to each other, okay? Which I'll call W0, W1, S0, and S1. W0 and W1 have the same fitness 1. S0 and S1 have the same fitness to each other, but they have a fitness 1 plus S, different from these two, and S is positive. So suppose again take, let's say, for illustrative purposes, I take these 4 samples. And initially, I have these 2 W0s and 2 W1s. Because S is a better one, it will likely to spread if it doesn't get lost. So perhaps an S came along, and then it spreads in the population, and at some point, it takes up over all the Ws. So what has happened? Initially, there was a diversity in zeros and ones. There were 2 zeros and 2 ones. Because of this spread of the selected, of this S locus, diversity is lost. Diversity is lost, and therefore this was, proposed as a mechanism for understanding why diversity may be, to understand the Levante's paradox, okay? Okay, so I want to understand this a little bit more, and therefore we again go back to this sort of model that I showed you earlier. Again, I have a large number of loci, and these are all single one will arise at each new locus and at different instants. The same sort of thing that I showed you earlier. Okay, but now they are asexuals. That is, if you like, these are interacting loci. Why interacting? Because you see, if this one gave rise to an offspring, S10, it gave rise to an offspring, then I cannot, these two have to be identical to the parent, right? So this cannot have an independent evolution at these two other loci. Okay, so this is the interacting loci problem, if you like. Okay, so here also when the S locus, when it spreads, then we have again some diversity left over, and we wanted to understand what does this SFS look like in this process. Okay, so because we are interested only in this S subpopulation when this S subpopulation is spread in the full population, so therefore we can just focus only on this S subpopulation. So therefore I'll erase all the W's. So but what happens there? You see S is increasing in time, the number of S, so it's like an expanding population size problem, okay? And that's the question that we can now look at. So therefore I'll modify the Moran process with growing size. So again, because we cannot do, we simply cannot, I mean not just we, I mean nobody can do the problem when you have interactions. So therefore again I'll assume independent evolution, but I have a slightly different Moran process. So let me describe that a little bit. So as before, we have this birth process and a death, but now the size of the population is not fixed. It can increase, and then as a result, no 0 has to die, maybe a 1 replicates. Similarly, if the size of the population decreases, a 0 dies, no other birth has to be given rise. Okay? So this is the microscopic model, the Moran process, in which I have a changing population size. Okay, so again, we would like to write down Fokker-Planck equation and look at the SFS, etc. You can do that, but you have to be slightly careful here. When we write down Fokker-Planck equations, these are for the fraction. That is something like little n divided by capital N, which is the population size. But remember here, denominator is also changing with time. So you have to be slightly careful, and if you do that a little carefully, then we arrive at this Fokker-Planck equation. So unlike earlier where I just assumed some n of t, s of t, here this is derived from this model in which n was fixed in time. And we find again this effective population size, which is smaller than the naive expectation. Okay, so if I now look at what this SFS looks like, it turns out it decays like 1 by x initially at small frequencies, and it has a different dependence, is 1 by x square. This dependence has also been seen in context of cancer, but at least in sweeps, I have not seen this come. Okay, so this is the simulations that Sachin did. Simulations are in blue for the asexual population that was interacting loci, and the answers assuming independent loci are in pink. So there is some discrepancy, but it's still a pretty good approximation. But what I'm really interested in, however, is not so much at the end of the process, but also the time dependence of F, which is under study, and so I will not describe it today. So I'll summarize with something very simple and general. So these are, so I'm trying to sell this to a lot of people actually. So there's a long history of population genetics, of exchange of ideas between population genetics, statistical physics, and probability theory. And Joachim has already explained some of them to you, and I've tried to do my bit. And if this kind of questions seem interesting, intriguing to you, I suggest you to look at this nice review article by Nick and others. Okay? The models that I described to you, this may be new to you, okay, some of them, but they are at the back of my mind is this resolving the Lewontin's paradox. Why is there a low diversity that what we actually see? Several factors have been identified, and I mentioned only a few, like demography, fluctuating selection, and other sweeps. But there are several factors, and people have generally looked at them individually and tried to see if we can explain the diversity patterns. Unfortunately, there's only a small change. We don't fully understand that still. And one suggestion from these authors is simply to look at multiple factors together. That's one suggestion, and another suggestion is simply this, and which looks at least more credible to me. The data that we are looking at, maybe they just have not reached equilibrium. They have not reached stationary state. Whereas when we are looking at data and understanding it, we are looking at assuming that it has reached stationary state. So therefore, one should actually look at the time dependence of this F and instead of the equilibrium answers. Okay? So that's what motivated us to look at these models with time-dependent rates, and we are interested in understanding the time dependence of SFS. Whether it helps or not, I don't know, but yeah, we can try. Thank you. Yeah.
Maybe this F, so it looks like that when you integrate over x, it diverges logarithmically or something, right?
Yeah, but it's, you have to integrate it not at the boundaries, excluding the boundaries.
So that
So
log
it's the
divergence is not.
No, it's this log n it will give. So you have to integrate with these finite.
Okay, and
Given
do you,
minus epsilons,
yeah, do you
yeah.
understand like why this divergence is coming from? I mean, like.
No, it has to diverge at very slow frequencies because I'm starting with very low frequency. Most likely it will get absorbed at the lower boundary. And therefore it will be spending a lot of time at the lower boundary.
Okay.
So it should, increase with the x, but why 1 by x and not 1 by x square or something? I don't know, it just comes out of the
Because like in
calculation.
the Fokker-Planck equation, it looks like a full derivative, so it's integrable on the right-hand side with respect to x, right? It just gives the two boundary terms.
Yes.
I mean, from the
Yeah,
Fokker-Planck
but
equation, it doesn't
it's
look
the
like
Green's
that.
function equation, right? This is the equation for the mu Sojourn time.
Yes.
Yes.
No, that Fokker-Planck equation you wrote down for f of x.
Yeah, but there's a boundary. So this equation is a slightly different model.
Yes.
Yes.
So even here you had this 1 by x times.
Yeah, so that's so this is the model, how to introduce new stochastic trajectories.
Okay, but
Yeah.
but this Fokker-Planck equation doesn't give you this 1 by x times.
Yeah, it does.
But this model doesn't look like there is any
No,
divergence
if I have
now.
S and N constant,
Okay.
it does give me the previous answers.
Okay.
Yeah.
So you're saying that if I just integrate this with respect to x, the right-hand side will give me divergence.
There's no divergence because this X has to be away from the boundaries.
But
There's no divergence.
But I can keep going closer to the boundary.
Yeah,
No,
but this still will get log. This is the integral that I have done here.
no, that previous that.
Yeah, this is exactly the same equation because they're independent trajectories.
Okay.
So it's essentially the same thing.
Okay.
So you will get just log n.
Okay.
Because of 1 by x, you'll just get log n. There's no divergence. Yeah. Yeah.
So first, a sort of simple technical question. So in the model of independent loci that you showed, the assumption is that all these mutations have the same selection coefficient. Is that right?
Yes.
Which is not really what you want, right?
Yeah,
I mean,
but yeah.
You
So
probably would
therefore,
like to have, distribution of selection coefficient, but then you cannot do anything.
We have
probably.
not taken care of, but I think other people like Bilger and so on, they have done that
Okay,
recently.
I
So it
see.
changes some, certain dependencies, there remains is the same. But yes, it's more realistic to look at the DFEs.
Yeah.
We haven't done that.
Right. The other about Leventin's paradox, I mean, I thought at least for humans, the story is always that the diversity that you see is essentially the number of people that got out of Africa, like 1 million, or not 1 million years, but in the migration out of Africa, that was like a severe population bottleneck, and that is what determines the effective population size.
Yes, so, yeah, so but like the numbers which the people have measured now is like 20,000 or so.
Yeah.
Maybe a factor of 5, so I mean.
Yeah, but this is not. It's not. This bottleneck scenario is not the whole explanation, or.
Probably not, because there will be some selection. So I don't think that, I mean, so that's the thing, right? We don't understand why the diversity is low. We don't understand, we don't have the quantitative understanding of those numbers. So we just know it's lower because of bottlenecks, but we don't know the numbers, no? So that's the whole problem, the 50 years old problem. So numbers we don't understand.
Other questions? If not, let's thank Javita
Okay.
again.
Thank you very much.
Now we have the picture downstairs.
Okay, so we just, before running to the lunch, we just go down and wait for the group photo, and then we go for lunch. Yes. So maybe we can just quickly go down. No discussion here. Like we'll have all the aggregation downstairs.